Hello and welcome, dear viewers. Today we will discuss about the primary structure of root in monocots and dicots. Root is generally considered as an organ in vascular plants that typically lies below the surface of the soil or which usually grows towards the gravity. But this is not always true about the root. A root can also be aerial as in epiphytes or aerating as in mangroves. A stem can also be underground, for example rhizome, tuber, etc. Here a question arises in our mind: how we can differentiate a root from stem? The answer is simple: roots do not bear leaves and are not differentiated into nodes and internodes as stem. Therefore, a root can be defined as a part of a plant body that bears no leaves and lacks nodes. Also in roots, the lateral branches develop endogenously, while in stem, the lateral branches develop exogenously. Roots are generally much more crooked or zigzag than stems, as their direction of growth is influenced by number of obstructions and moisture content of the soil. Roots are present in almost all the vascular plants with few exceptions, such as Utricularia, which is an aquatic insectivorous plant. A true root system consists of a primary root, also called main root, and secondary roots, also called lateral roots or branch roots. On the basis of the origin, root system can be divided into following two subcategories: taproot system and adventitious root system. Taproot system consists of the primary root formed by direct elongation of the radicle and its lateral branches. The primary root or taproot normally grows vertically downward to a shorter or longer depth, while its lateral branches like secondary branches, tertiary branches, etc. grow obliquely downward or in many cases spread horizontally outwards. This type of root system is present in most of the dicots and gymnosperms. Adventitious root system. It includes those roots which develop from any part of the plant other than radicle. This type of root system is mostly found in monocots, where the radicle is short-lived. These roots originate from different plant organs such as stem, leaf, etc. Fibrous roots in monocots, foliar roots of Bryophyllum, tuberous roots of sweet potato, and prop roots of banyan tree are some of the examples of adventitious roots. Functions of root. The major functions of root are absorption of water and inorganic nutrients, anchoring of the plant body to the ground, and storage of food material as in carrot, radish, turnip, etc. The roots or parts of roots of many plant species have become specialized to serve adaptive purposes like propagation of parent plant as in sweet potato, respiration as in mangroves, photosynthesis as in Tinospora, and buoyancy in many hydrophytes. These functions of roots other than primary functions are known as secondary functions of root. The respiration in mangroves is carried out by specialized roots known as nematophores. Nematophores are the specialized spongy roots developed by some swamp plants or halophytes which extend above the water surface and enhance the gas exchange between the atmosphere and the subsurface roots to which they are connected. The roots of most leguminous plants are associated with the beneficial microbes like nitrogen fixing bacteria and help in the atmospheric nitrogen fixation. The presence of these bacteria results in the formation of nodules on the root. Roots also have important role in preventing soil erosion by holding the soil particles together and making them resistant to erosion by wind or water. Zones of root. A root is generally divided into 4 distinct zones depending upon the degree of maturation and activities taking place in each area. These regions, starting at the tip and moving upward towards the stem, are the root cap, zone of active cell division, zone of cell elongation, and zone of maturation. Root cap or zone of protection. The root cap is a cup-shaped, loosely cemented mass of parenchymal cells that covers the tip of the root. It is quite large in some plants, while in others it is invisible or absent. The cap is a unique feature of roots. The tip of the stem has no such structure. From its shape, structure, and location, its primary function seems clear. It protects the cells under it from abrasion and assists the root in penetrating the soil. Phenomenal number of cap cells are produced to replace those worn off and lost as root tips push through the soil. The movement is assisted by a slimy substance called mucigel, which is produced by cells of the root cap and epidermis. It has been observed that the root without root cap seems to grow randomly instead of more or less downwards, suggesting that the root cap also functions in the perception of gravity. Zone of cell division. A mass of actively dividing cells lies under and behind the root cap called zone of cell division or root apical meristem. Like the stem apical meristem, it produces the cells that give rise to the primary body of the plant. Unlike the stem meristem, it is not at very tip of the root. It lies behind the root cap. The parenchymal cells of the meristem are small, cuboidal, with dense protoplasts and devoid of vacuoles, and with relatively large nuclei. Between the area of active division and the inverted cup-like cap is an area where cells divide more slowly. This region is called quiescent center. Most cell divisions occur along the edges of this center and give rise to column of cells arranged parallel to the root axis. The apical meristem of the root organizes to form the 3 primary meristems, namely protoderm, which gives rise to the epidermis, procambium, which produces xylem and phloem, and the ground meristem, which produces the cortex. Zone of cell elongation. The region of elongation which merges with the apical meristem usually extends about 1 centimeter or less from the tip of the root. The cells in this zone stretch and lengthen as small vacuoles within the cytoplasm merge and fill with water. 1 or 2 largely vacuoles occupy almost all of the cell volume, up to 90% in fully elongated cells. Here the cells become several times larger than their original length and also somewhat wider. Cellular expansion in this zone is responsible for pushing the root cap and apical tip forward through the soil. Zone of maturation. Most of the cells mature or differentiate various distinctive cell types of the primary tissues in this region, which is sometimes called region of differentiation. It is easily recognized because of the presence of numerous root hairs that extend into the soil as outgrowths of single epidermal cells, therefore also called root hair zone. Internal organization of the root. The root shows comparatively simple internal organization than stem due to the absence of nodes and internodes and lateral appendages. 3 zones can be distinguished in a root in the primary state of growth. Number 1 epidermis, number 2 cortex, number 3 vascular system. Epidermis, also called epibellima. The epidermis of root is a continuous layer composed of tightly placed, thin-walled, barrel-shaped uncutinized cells. In some herbaceous perennials, where epidermis is long-lived and act as chief protective layer, the outer tangential walls of the epidermis cells are cutinized. Epidermis is usually uniseriate, but area roots of some orchids and epiphytic aroids possess a multiseriate epidermis called velamin. The velamin is composed of compactly arranged non-living cells which often have secondary wall thickenings. It is primarily an absorptive tissue, but it is believed that velamin is basically a mechanical tissue which protects and checks loss of water from the cortex. The cells of velamin are filled with air in dry season, but in rainy season they are filled with water. The epidermis of young roots bear innumerable unicellular root hairs which are adapted for efficient uptake of water and minerals. The root hairs are initiated a few millimeters from the root apex in the region of maturation. The average lifespan of root hairs is only 2 to 4 days. As the root grows, hairs present on the more mature portion usually die and dry out, and new hairs continue to develop in relatively young parts of the root. Occasionally, roots are persistent in certain herbaceous plants such as Eupatorium purpureum. All the epidermal cells of root are capable of initiating root hairs, but only a few of them do so. The cells of epidermis which form root hairs are sometimes morphologically distinct from other cells. These cells are known as trichoblasts. A root hair originates as a bump on the epidermal cell, and this bump has cytoplasm and nucleus towards the tip. Later the bump extends and often becomes several millimeters long, and the cytoplasm spreads all along the wall in the form of a thin layer enclosing a large central vacuum. The root hairs may be regarded as the structure meant for increasing the absorbing surface of the root. It should be noted that absorption of minerals through the root hairs is active and selective process. In aquatic plants, root hairs grow straight at right angles to the root, but in plants growing in soil, they assume various angles depending upon their contact with soil particles. Cortex. The cortex of root is much simpler than that of stem. It is usually composed of thin-walled isodiametric parenchymatous cells with many intercellular space. Collenchyma is usually absent in root cortex, but sometimes fibers, sclerids, idioblasts, and latex cells may occur. The cortical cells are usually devoid of chloroplasts, but they store starch. However, in roots of some aquatic plants, for example Tinospora, and in aerial roots of many other plants, few outer layers of cortex contain chloroplasts and perform photosynthesis. The cells of cortex are usually arranged irregularly, but sometimes they are arranged in distinct radial rows like Avena sativa, or in concentric layers as in Rapetia. In aquatic plants, large air cavities are formed due to the degeneration of some of the cortical cells. These spaces are mostly filled with air and help them in buoyancy. In most dicotyledons, the cortex is replaced by suberized cells as a result of secondary growth. But in some herbaceous dicotyledons which lack secondary growth, it is permanently retained and develops various types of mechanical tissues. The cortical cells in roots of many gymnosperms and some dicotyledonous species, for example the species of Rosaceae and Brassicaceae, have reticulate or band-like thickenings which may be lignified. In many monocotyledons and few dicotyledons and gymnosperms, 1 to several subepidermal layers of cortex differentiate into protective tissue called exoderms. As in gram roots, the cells of exoderms have suberized walls and contain protoplasmic contents. Endoderms. The endodermis delimits cortex from stele is of universal occurrence in the roots. It consists of a single layer of closely packed living cells. The endodermal cells are characterized by the presence of thickenings called Casparian strips in their radial and tangential walls. These thickenings are in fact an integral part of primary wall and middle lamella, and they are formed due to the deposition of suberin and lignin. In roots with secondary growth, the endodermis is usually cast off together with the cortex, but in roots which remain in primary state, a suberin lamella is usually formed over the whole inner wall of the endodermal cells. Some of the cells of endodermis, usually the ones opposite to the protoxylem, remain thin-walled and are called passage cells. They allow radial diffusion of water. Various functions has been assigned to the endodermis, and it has been considered as a mechanical protective layer, a sort of inner accessory epidermis, a layer concerned with the maintenance of the root pressure, and an airtight layer preventing the water conducting cells from being clogged with air. Recent studies have shown that endodermis is a sort of barrage that prevents the leakage of nutrients from vascular tissues and regulates the inflow of water with mineral salts absorbed by roots through the cortex directly into the xylem elements. Pericycle. The pericycle, which occur beneath the endodermis, constitutes the outer boundary of the primary vascular cylinder of the roots. It is usually a complete ring of cells, but sometimes its continuity is interrupted opposite the xylem elements, as in Cyperaceae and Graminae, or opposite the phloem elements, as in Potamogenaceae. Generally, the pericycle consists of one or more layers of thin-walled parenchymatous cells, but a multiseriate thick-walled pericycle occurs in Salix and Smilex. In some parasitic dicotyledons and a few aquatic plants, pericycle is absent. In the pea root, pericycle is one cell layer thick opposite the phloem and 3 to 4 layers opposite the xylem. Pericycle differentiates quite earlier in the histogenesis of root and is derived from the procambial strain. It retains its meristematic characteristics. The pericycle functions as the site of origin of lateral roots, but in roots which show secondary growth, cork cambium, which we also call the phallogen, and portions of vascular cambium develop from the pericycle. Vascular tissue. The arrangement of primary vascular tissues, means xylem and phloem, is different in the root as compared to the shoot. In the root, xylem and phloem lie on different radii, that is, they are radial in arrangement. In the dicotyledonous roots, there are usually 2 to 6 groups of xylem and an equal number of alternating phloem groups. In the monocotyledonous roots, their number varies from 8 to 20. Depending upon the number of xylem bundles, roots are called monoarc with 1 xylem and phloem bundle, diarc with 2 bundles, triarc with 3 bundles, tetraarc with 4 bundles, pentaarc with 5 bundles, or polyarc with many bundles of each xylem and phloem. The vascular tissue differentiates from the procambium. The first elements of xylem are the most peripheral, and the subsequent differentiation proceeds towards the center of the root. That is, new vessels are added towards the center. Such an arrangement of vessels is known as X-arc condition. In situm, the case is reverse, that is end arc, where direction of vessel differentiation proceeds from center to periphery. The small peripheral elements of the xylem with spiral or annular thickenings are formed during the initial elongation of the root and are known as protoxylem elements. The wider and more central elements with pitted or scaliform thickenings form the metaxylem. The phloem also differentiates centripetally, and the first phloem is recognizable even before the first xylem. The primary phloem of the root consists of sieve tubes, companion cells, and phloem parenchyma. The first formed phloem elements, that is the protophloem, are not associated with companion cells, but the later formed elements, meaning metaphloem, are commonly associated with the companion cells. Pith. The pith is usually absent in dicot roots, but present in monocot roots and occupies very small area in the center of the root. It consists of thin-walled parenchymatous cells with intercellular space. Sometimes due to the development of metaxylem vessels in the center, pith is completely obliterated. Let us take the example of maize root, a typical monocot root. A transverse section passing through the maize root reveals the following details. Epibellema. It is the outermost covering of root formed by a single layer of compactly arranged, thin-walled, barrel-shaped parenchyma cells involved in absorption of water. A cuticle and stomata are absent. Some of the epibellema cells are produced into long unicellular projections called root hairs. Hence, epibellema is also known as pelliferous layer. Cortex. Cortex is major component of the ground tissue of root in maize and is represented by several layers of loosely arranged parenchyma cells. Intercellular spaces are prominent. The cortex is mainly meant for storage of water. The cells also allow the free movement of water into the xylem vessels. Endodermis. It is innermost layer of cortex formed by compactly arranged barrel-shaped cells. Some of the cells in the endodermis opposite the protoxylem are thin-walled, known as passage cells. The passage cells allow water to pass into the xylem vessels. The remaining cells in the endodermis are characterized by the presence of the thickenings on their radial walls. These thickenings are known as Casparian thickenings as mentioned earlier. The Casparian thickenings play an important role in creating and maintaining a physical force called root pressure. Pericycle. Pericycle is the outermost covering of the stele represented by a single layer of parenchyma cells. Pith. It is the innermost region of the root representing the central axis. It is composed of few loosely arranged parenchyma cells. Vascular bundles. Vascular bundles are radial in arrangement. There are 8 bundles each of xylem and phloem. Hence the condition is described as polyarc. Xylem is described as X-arc, means protoxylem is towards periphery and metaxylem is towards center. If we look at the anatomy of sunflower root, a typical dicot root, a transverse section passing through the root reveals the same story as in maize, except the vascular bundles and pith. The vascular bundles are tetrarch in sunflower, that is, there are 4 bundles each of xylem and phloem occurring alternately. Pith is absent in the older roots of sunflower. Development of lateral roots. In contrast to the lateral appendages of the stem, which originate exogenously from the shoot apical meristem, lateral roots are initiated endogenously behind the apical meristem, typically in the region of maturation. The lateral roots of both gymnosperms and angiosperms are usually initiated in the pericycle opposite to the xylem ray and subsequently grow through the cortex. The position of the lateral root development varies depending upon the position of vascular strand in the parent root. In diarch root, it arises between the phloem and xylem strands. In tri, tetra and pentaarch roots, opposite the protoxylem, and in polyarch roots, opposite to the protophloem strands. Primordial of lateral roots are formed just behind the region of root hairs by periclinal divisions in the pericycle, followed by divisions in both periclinal and anticlinal planes. The young lateral root grows through the cortex and epibellema, that is epidermis, of the parent root and passes into the soil. During this process, the cells of the cortex are crushed and pushed aside. The passage of lateral roots through the cortex is mainly by mechanical penetration, but enzymes may also facilitate the growth of lateral roots through the cortex. Root cap and promeristem of the growing lateral root develops when it is settled within the cortex. In mature roots, where pericycle has been slowed off, lateral roots commonly initiate in the cambium. This is all we have in today's lecture. Thank you for watching.